Okay, good evening, friends. Today we want to talk about another topic in hematology. Okay, hematology. So, the topic tonight is hematological malignancy. Hematological malignancy. It is very important as a scientist or as a medical lab student or health professional to know more into hematological malignancy. Okay, is very important, especially the lab technicians, because their diagnosis and care of the patient basically centered in the lab. So as a medical lab science student, you must know and understand the concept of hematological malignancy. So with this lecture, we will look critically at we talk about some introduction, and then the various types of hematological malignancies. There we will also talk about the lab diagnosis. That is my key concern, the lab diagnosis. How are we able to diagnose hematological malignancies? Very, very important. So a lot of topics. Let's continue. Okay, good. So hematological malignancies, before we understand hematological malignancies, okay, first of all, let's talk about, or let's look at some little bit introduction. Okay, it's very, very important. So talking about hematological malignancies, okay, now in the lab, hematological malignancies can be a little bit confusing, okay, because in most cases, if you don't take care as a medical lab scientist, you confuse yourself with some causes to hematological malignancy, because they all present with the same symptoms, okay, the same symptoms. That is why I want us to look at some basic causes, okay. For instance, when you take a sample from a patient and you run your full blood count, okay, you may see that there is, let's say, thrombocytopenia, there is thrombocytosis, possibly there is low level of hemoglobin. You can also see that there is a leukocytosis. Now, all these things are blood cells that are, either decreasing or increasing, okay. Now, with respect to hematological malignancies, we focus much on the WBCs, okay, because they really affect the WBCs, okay, the blood cells. Not only WBCs, but then basically white blood cells are affected, okay. So talking about hematological malignancies, okay, I want us to first look at some, causes of increased level of WBCs, okay, before we understand or before we go into hematological malignancies. Now, WBC, okay, we have disorders that can cause increased level of WBC, that is the white blood cells, okay. So other than hematological malignancies, we can have other causes that can cause increased level of what? WBCs, okay. Now the first one I'll talk about is let's put it like this. Proliferative disorders of WBC, that is factors that causes high levels of or increased level of white blood cells, okay. So they can be grouped into two. So proliferative disorders of WBCs can be grouped into two, okay. The first one that we talk about is reactive proliferative disorder of WBCs, okay. So you can have the reactive proliferative disorder of WBCs. Okay, I'll talk about that. And we can have the neoplastic proliferative disorders of WBCs. The neoplastic, okay, is actually what we're going to talk about into details. That is the malignancies, like the hematological malignancies. Now, what does it mean to say something is what does it mean to say there is what is called reactive proliferation? Okay, so reactive proliferation occurs, let me highlight it. Reactive proliferation occurs when WBC's numbers increase due to an inflammation or challenge to the immune system. You understand that? So it means that in most cases, okay, in the body there can be inflammation, and you know these inflammations can be caused by bacteria, they can be caused by other organisms, okay, any foreign antigen, and most at times, or in most cases, this WBC's level increases. Okay, so it is very important to know other factors that can cause increased level of WBCs, so that in your attempt to diagnose somebody with hematological malignancy, let's say leukemia, you don't confuse yourself. Okay, maybe the person high level of WBCs may be possibly due to this inflammation or some challenge to the immune system. Okay, so you don't confuse yourself with the two. Good. So when I say reactive proliferations, they actually occurs when WBCs numbers increase due to what? Inflammation or challenge to the immune system. Very, very important. Okay, very, very important. Good. Now, WBC numbers increase in peripheral blood in reaction to inflammation or infection or stress or trauma. Okay, so very, 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 very important. Very, very important. WBC numbers may increase in peripheral blood in response to inflammation or stress. In this case, the increased level of WBC is being caused by other factors, you understand, other than leukemia or any other malignancy. Okay, now let's focus on the reactive proliferation, the factors that can cause increased level of WBC. One is leukocytosis, and another one that we talk about is leukemoid reaction. Okay, so therefore, what is leukocytosis? What is leukemoid reaction? Now, let me take you back. You know that the normal WBC count in blood is range from 4,000 to 11,000 microliter. Okay, 4,000 to 11,000 microliter. Okay, and it is very, very important to understand the values, so that when you are working in the lab, if you know the range, and any time there is increase or decrease, you know that there's a problem. Okay, now, so the normal WBC count is 4,000 to 11,000. Okay, now whenever there is WBC count, which of course is less than 4,000, it means there's decreased level of what? WBC. In that case, you say you have leukopenia. So leukopenia is low level of what? WBC. That one is clear? Good. And of course, when there is increased level of WBCs above the higher point or the upper limit, which is 11,000 microliter, you say you have leukocytosis. In this case, there is increased level of WBCs. But one thing is that the reaction is proliferative. It's not as a result of malignancy, it's proliferative. Okay, very important. Now, in leukemia reaction, even though there is a relative leukocytosis, the increased level of WBCs is very, very, very high as compared to the proliferation or the leukocytosis. So, in leukemia reaction, you see that the WBC level is very, very, very high. Can you see that? 30,000 to 50,000, very, very high. Unlike the leukocytosis, which is above 11,000, 12, can be 12,000, 13,000, but this one is 30,000 to 50,000. So in leukemia reaction, there is a remarkable increase of WBC level. So take note of that. Okay. So here we've established the fact that increased level of WBC is leukocytosis, which can be as a result of reaction, which is induced by inflammation to the immune system, whatever it is. Okay, and we say that in leukocytosis, the WBC increases above the upper limits. Okay, more than 11,000. But in leukemia reaction, even though there's reactive leukocytosis, but the increase in WBC becomes very, very high. Okay, very, very high. So we proceed. Now, leukocytosis, there are different forms of leukocytosis. Okay, of course we can have neutrophilic leukocytosis, which we normally call the neutrophilia. Okay, so you run your full blood counter, you see there is increased level of neutrophils, that is neutrophilia, and in that case it may be due to bacterial infection. Okay, in most cases bacterial infection. In most bacterial infections there is a high level of neutrophils. Okay, good. Now you can also have eosinophilia, which is normally seen in bacterial infection. Then in most cases you can have basophilic leukocytosis, which is called basophilia. That one is in allergic reactions. Okay, allergic reaction. Of course, lymphocytosis seen in cases like tuberculosis, and in chronic inflammation you can see monocytosis. So what am I trying to do? I'm trying to draw attention that running full blood counts, seeing leukocytosis can be as a result of some reactions to the immune system. Okay, which is not actually as a result of malignancy, and these reactions are called proliferative disorders of WBC. And of course it can be infections by bacteria, which is neutrophilia, and so on and so forth. Okay, so in leukemia, reactive leukocytosis and leukemia reactions. Okay, in both leukemia, reactive leukocytosis and leukemia reaction, WBC levels may rise, and it becomes very important to differentiate them. Okay, that is why I am going back to these things. Okay, good. So very, very important. Now let's go back to hematopoiesis. Okay, now I'm done with reactive leukocytosis. I'll talk about the malignancy. Okay, but before that, let me quickly go to hematopoiesis or hemopoiesis. Okay, good. So talking about hematopoiesis, I hope you remember. See the structure, it reminds you
I
of
can't connect at the moment. Try again in a little bit.
So this one reminds you of something. This is called a bone marrow. Bone marrow. That is where hematopoiesis occur. Okay, very important. Now, hematopoiesis is the production of blood cells. Okay, production of blood cells, and that normally occurs. It normally occurs in the bone marrow, like I showed you. Okay, in the bone marrow, very, very important. Now, In the bone marrow, we have something we call the hematopoietic stem cell. Poietic stem cell. Now, this hematopoietic stem cell is basically the cell that generates into different types of cells, which result in production of blood cells. Okay, so hematopoietic stem cell can be divided into common myeloid progenitor cell, and we can also get a common lymphoid progenitor cells. Okay, so let's talk about them one after the other. Now, the common myeloid progenitor cell lineage, coronae-forming unigem, the common myeloid progenitor cells, okay, leads to the production of some blood cells. Okay, basically you can get the blood cells at the myeloblast, promyelocyte. These are the cells that leads to the generation of the matured cells. Okay, of course you have the myeloblast, promyeloblast, myelocyte, and the metamyelocyte. They are all blood cells. Okay, blood cells. And remember that all these blood cells are not supposed to be seen in the peripheral blood. Okay, so we have established the point that hematopoietic stem cell is in the bone marrow. Okay, and inside the bone marrow, we have hematopoietic stem cell, which can be divided into common myeloid progenitor cell and then common lymphoid. Of course, we said the common lymphoid, we can have the blood cells, the immature cells. Okay, immature cells, which are the myeloblast, promyelocytes, metamyelocytes, and then the myelocyte. Okay, very important, very important. All these cells give rise to the granulocytic cells, and these granulocytic cells normally are the band cells. Okay, you can get the band neutrophils, the band eosinophil, the basophil. They are the matured cells, and they are supposed to be seen in peripheral blood of any healthy person. Okay, good, very important. So the common lymphoid, so we've spoken about the common myeloid. Okay, the common myeloid, which give rise to the myeloid cells. Okay, which are the neutrophils, eosinophils, and the rest. Now, the common lymphoid progenitor cells further differentiate into lymphoblasts. Okay, lymphoblasts. So the blood cells. Now, these lymphoblasts will then differentiate into genetic profiles, which becomes naive B cell, which we normally call a B lymphocyte. It is naive because it is assisted. It hasn't been exposed to any antigen, so it doesn't know anything. Okay, good. So naive B cell, once it is exposed, it starts fighting against foreign antigens. So they play a major role in the immune system. Okay, remember you have adaptive immune system and innate immune system. Okay, so these cells are very important when it comes to the immune system, the function of the immune system. Okay, good. So the pre-T cell, we are the T lymphocyte, and the natural killer cells. Okay, they are very important, very important. So this is a picture showing the cells, how they are produced in the bone marrow. Okay, they are produced in the bone marrow. So we have the hematopoietic stem cell here, which is the main stem cell. Okay, now it is being differentiated into common myeloid and then the common lymphoid. Okay, now from the common myeloid we can get the erythrocyte, which is the red blood cells. We can get the myeloblast, which would then differentiate into the mature cells. So we have the basophils, we have the neutrophils, eosinophils, and monocytes. Okay, these are mature cells, and they are all coming from the common myeloid progenitor cells. Now, the common lymphoid progenitor cells give rise to the B lymphocyte, T lymphocyte, and the plasma cells. Okay, remember that this is what actually occurs in the bone marrow, so that we will get accurate cells, mature cells. Okay, this process is well regulated. Okay, it's about a cell cycle. The cells must pass through the cell cycle, mitosis occur, and they give rise to all these cells, and it is well regulated. Now, the genesis of hematological malignancy basically occurs when there is either mutation to the regulatory process, the factors that regulate this process. So when that happens, you see that all the malignant cells will push away all these hematopoietic stem cells from the bone marrow, and they will start forming clones. And so we are not going to get our neutrophils, our basophils, our mature cells in the peripheral blood. What we may be seen is the immature cell, the blood cells and the lymphoblast cells. So very important to understand this concept. Very, very important. So the process of hematopoiesis is regulated by many factors. Like I said, the cell cycle is regulated in such a way that no malignancy occurs. The hematological malignancies occurs due to clonal expansion. Now, when we say clonal hematopoiesis, is defined by the overrepresentation of single clone in blood or bone marrow. So, in the bone marrow, instead of the hematopoietic stem cell to function to give rise to normal cells, there will be mutation. Mutations. And when there is mutation, there is a clone. The cancer cell form clones, and they'll push away the main cells. Main cells, and they will take control of the bone marrow and give rise to malignant cells, which is very bad. And that is the genesis of hematological malignancy. In this case, remember that we are no more going to see matured cells, but we are going to see malignant cells. So an important feature of malignancy is clonal expansion. So in most hematological malignancy, the cancer cell uses all the nutrients, and so I've spoken about this. Now, in hematological malignancy, two key genes are mutated: the oncogenes and the tumor suppressor genes. I'll talk about them very soon. What is oncogene? What is tumor suppressor gene? Very, very important. Very, very important. So in the lab, these are some of the things that we can use to investigate hematological malignancy. The light microscope, which is very common in developing countries, like Ghana, we focus much on the light microscope. Flow cytometry, this is very important, when it comes to the diagnosis of hematological malignancies. But this is very expensive. Not all labs, but when you go to some countries, they have it. Karyotyping, very important. I'll talk about these things in the diagnosis of hematological malignancy. Forensic in situ hybridization, fish technique, very important. DNA microarray, a lot of them there, so talk about them. So that if I ask, so let's talk about oncogenes, tumor suppressor genes, and telomeres. What are their roles in hematological malignancies? Very important to know this. So oncogenes. Basically, oncogenes are derived from proto-oncogenes, which is activated by gain of function mutation. Now, gain of function mutation is sometimes there is a point deletion or point mutation. There is insertion, there is deletion, and translocation or inversion of specific chromosome. So these proto-oncogenes, physiologically, they involve in regulating signals that activate gene expression. So if hematological stem cell is to express into, let's say, common myeloid, common lymphoid, to give rise to mature cells, these oncogenes play these proto-oncogenes play major role. Now these proto-oncogenes can be mutated You understand? Yeah. So when they are mutated by, let's say, gain of function mutation, either by point mutation, by insertion, or by deletion, they give rise to what? Oncogene. Okay, oncogene. And when they give rise to oncogene, this oncogene will lead to cell division, and thus malignancy, which is very important. So proto-oncogenes does not cause malignancy, okay, but when it is being activated into oncogenes, the oncogenes will rather cause cell division, okay, to facilitate cell division, and may lead to clonal expansion and therefore malignancy. Okay. Now, oncogenes have been detected in about 15 to 20% human tumors. Okay. The most common etiology is chromosomal aberration, like I said, mutation. So there is mutations in chromosome, and it can be point mutation, insertion, and deletions. Sometimes translocation or inversions. Okay, very very important. So remember that proto-oncogenes are there to regulate normal hematopoiesis. Okay, in that they regulate signals that activate gene expression. Okay, when they are activated into oncogenes, this oncogene will facilitate cell division, which will lead to malignancy. Okay, so oncogenes, which is driven by gain of function mutation, causes hematological malignancy. Now, tumor suppressor gene. Tumor suppressor gene. Tumor suppressor gene is very very important. Very very important when it comes to the generation or the maintenance of hematopoiesis. Okay, now the tumor suppressor gene, its function is to suppress cell division or promote apoptosis. So if there are some malignant cells that are dividing uncontrollably, these tumor suppressor gene are there to suppress and promote apoptosis. You know, apoptosis is where there is a programmed cell death. Okay, so through the cell cycle, some cells are not obeying some principles, like they want to pass through the checkpoint and misbehave. So there will be programmed cell death, apoptosis, and this one is caused by the tumor suppressor gene. Okay, tumor suppressor gene, very important. And of course, this tumor suppressor gene, when there is loss of normal chromosome or genes, may lead to reversion to malignancy. Okay, so if there is loss of function, they can cause malignancy. Not gain of function, loss of function, malignancy. And this is achieved when there is loss of function mutation, like I said that. Okay, I said that. Very 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 important. Very 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 important. So